Bismillahirrahmanirrahim. Let's talk more about different electron donors and acceptors which are used in different metabolic processes that are involved in biodegradation. Let's talk about some more examples and exceptions as well. So we were talking about de-halo respiration that can be performed by different microorganisms, but it does not require that the dehalogenated compound also serve as a source of hydrogen. हमेशा ऐसा नहीं होता. There are so many different electron donors and acceptors which can be used. If a different organic compound is the initial electron donor, then competition can be there for the available hydrogen, and it can be quite interesting as well. For example, we can see in this sketch, let's suppose if the electron donors are basically mixed complex organic materials, it starts from the hydrolysis leading to the generation of organic monomers. These monomers can then be fermented to produce different alcohols, acids, and also acetate and hydrogen. Now, there are so many different microbial populations which are present in this environment, for example, and there are so many other electron acceptors as well. You can see here sulfate, iron, carbon dioxide, and there are so many halogenated compounds as well, which can be dehalogenated by different microbial organisms. So there tends to be a competition to get the electrons which are present in the acetate and hydrogen. Sulfate reducing bacteria will compete with other bacteria to get these electrons in order to reduce the sulfate to sulfide, and so will the bacteria that can do it with iron and carbon dioxide as well, and different bacteria and microorganisms which are involved in dehalogenation as well. So in a complex environment where the microbial communities are complex and where the chemical compounds are complex as well, there always occur a competition for the availability of nutrients and for electron donors as well, and depending on who succeeds, different types of compounds will be reduced more efficiently as compared to other compounds. Now this also illustrates that while reductive dehalogenation can be very important in transformation of halogenated organic compounds, it is not something that can be assumed to occur automatically. Obviously automatic reaction नहीं है, इसमें different competition involved है. Moreover, इसमें thermodynamics involved हैं that should also be considered. Now, when we talk about this conversion, microbes, they tend to be quite specific in the transformation of compounds. वो normally specific होते हैं towards the target compounds. For example, the organisms that can dehalogenate M-chlorobenzoate may be different from the organisms that can dehalogenate O-chlorobenzoate. So you can see here the microbes can be quite specific depending on the basically specificity of their enzymes. Now the microorganisms that can dehalogenate tetrachloroethene generally are different from those that can dehalogenate dichloroethene. So you can see here, depending on different types of compounds, depending on slight variations in their chemical structures, the compounds that act on these the microbes that act on these compounds can be different. So this was about something about different electron donors and acceptors and how this process can happen and how the exceptions can be there. I will see you in next lessons.